Zero the spectrophotometer with deionized water. Measure the OD600 values by using 3 milliliters of your culture. Make sure to clean the outside of your cuvette with a Kim wipe before placing it in the spectrophotometer, as failing to do so can result in inaccurate OD readings. You will perform step 2 4 times, once for each different treatment condition. Make sure to record these values for later use. In steps 3 and 4, you will pipette the 3 milliliters of each culture used to measure the OD600 values into 2 marked 1.5 milliliter centrifugation tubes. You will then centrifuge each tube. It is important to make sure that the tubes are balanced. You will know if they aren't, as you will hear a loud noise. Note that you will have more than the 2 centrifuge tubes shown here. Also, double check that the centrifuge lid is on before starting. This is very easy to forget. Carefully dump the supernatant out of the tube into the liquid waste containers on the bench, leaving only a pellet of cells at the bottom. Pipette the 1 milliliter of resuspended cells into the other tube containing the other pellet. Pipette up and down 5 times to suspend the other cells. Here, the transfer of a resuspended pellet from one tube to the other is shown. Centrifuge for 1 minute, following the same instructions discussed for step 4. Use a P1000 to remove as much of the supernatant as is possible without disturbing the pellet. Start to boil water for incubation later. Suspend the cell pellets in 50 microliters of chilled lysis buffer for every 1 milliliter of culture at an OD600 of 1. Pour glass beads found in a separate centrifugation tube on the bench into the centrifugation tube with your yeast and lysis buffer. Transfer all of the supernatant, which contains the protein, to a new centrifugation tube. Add the same volume of Lamley buffer as you did lysis buffer. Make sure to adjust different volumes to each lysate since it is based on OD readings. Incubate the samples in boiling water. The following segment of this video will demonstrate how to correctly set up your gel for an SDS page. Note that you will probably only be running one gel. Where another gel is being placed in the video here, you will be placing a buffer dam. Add running buffer to the gel box up to the gel cassette's bottom green gasket. Now you will load your samples into the wells of the gel. There is a table in the lab manual we recommend filling in to keep track of what you've loaded into each well. Use a P20 to pipette 12.5 microliters 2 times for a total of 25 microliters of each sample into individual lanes of the gel. You will add only 10 microliters of ladder. Place the tank lid with power cables onto the top of the buffer tank and plug the power cables in. Red matches with red and black with black. Activate the power box and start your electrophoresis. Every 10 minutes, check your gel to make sure that the samples have migrated out of the lanes and farther down the gel. Note that there will only be one ladder per group, unlike in the gel shown here. The blue dye should migrate to within about 1/4 inch from the bottom of the gel. Soak foam pads, filter paper, and nitrocellulose paper in separate containers, each filled with transfer buffer in preparation for the next steps. When 30 minutes have passed and you're satisfied with the migration of your samples, disconnect your setup and pour the buffer into a used buffer container. Pry the plastic gel plate off of the cassette. There are 2 tools on the bench for this purpose. You can also use your fingernail. Cut the top lanes away with a spatula or scalpel. Don't forget to make a notch at the top of your gel so you can remember where lane 1 is. Gently transfer your gel to the UV box. Expose the gel to UV light for 2 minutes. Now take a picture of your gel. making sure that the camera settings are correct as outlined in the lab manual. Now you will make your sandwich. Be careful with this, as orientation is very important in these next few steps. There's a useful image in the lab manual to help you. Your cooling tub may either contain cold water or ice packs, depending on what is available. Fill the buffer tank with transfer buffer up to the lip. Again, make sure to match red with red and black with black. Now you will take apart your sandwich. You'll need to make a notch on the membrane, as we did with the gel. Make sure your protein ladder has transferred to your nitrocellulose. If not, something may have gone wrong. If you shared a membrane with another group, you can now divide the membrane using a scalpel and proceed in individual groups. Your membrane should be placed in the petri dish with the side that faced the gel facing up in the container. Pour TBST plus 5% milk into a square petri dish. The volume does not have to be exact, but should be sufficient to just cover the membrane, about 10 milliliters. Place the lid on the petri dish and put the dish on the rocker for at least an hour. You can either incubate overnight or move on with the next steps right away. Slowly pour the TBST and 5% milk into the sink, such that the membrane does not fall out of the dish. Don't worry, it sticks well. Cover the membrane in deionized water and swirl to wash. Repeat the water wash one more time. Dilute stock primary antibody, anti-beta-galactosidase, by pipetting 2 microliters into a 15 ml tube. Aliquot tubes can be found in the freezer. Pour the 10 ml of diluted secondary antibody solution into a dish containing your membrane. Incubate the membrane on the rocking platform at room temperature for 1 hour. Pour enough TBST plus 5% milk into the dish to just cover the blot, about 5 ml. Put the dish on the rocker for 5 minutes. You'll then pour the TBST plus 5% milk into the sink and repeat this step 2 more times. This washing step removes the unbound primary antibody and blocks the nitrocellulose again to limit nonspecific binding of the secondary antibody. Dilution of the secondary antibody isn't shown here, as it is quite similar to the primary antibody and is outlined very well in the lab manual. Add 10 milliliters of Western blue detection reagent to your dish. Watch as the protein bands develop. Focus on the area of the membrane where you expect your protein of interest to be, about 120 kilodaltons. It is possible to overexpose the membrane, so be ready to stop the reaction once the bands start to get dark. Shown here is what your nitrocellulose should look like. Don't forget to scan your membrane for later analysis.